Thank you for joining us at this BNA Credibility Lunchtime webinar. My name is Cassandra, my pronouns are she, her, and I'm the Open Science Community Engagement Coordinator at the Wellcome Centre for Integrative Neuroimaging at the University of Oxford, WIN Centre. And I'm very pleased to welcome you all and also welcome our speaker today, Roni Tibon. Roni is an assistant professor at the University of Nottingham in the School of Psychology, and she is working on neurocognitive mechanisms of memory over the lifespan. Roni has done postdocs in Cambridge and Israel, and she's been a champion of pre-registration for a long time. Pre-registration posters, which are a feature of the BNA Festival, which is happening in April. You can still submit your posters today. And Roni's also co-authored papers on good scientific practices with Meg, with specific recommendations for pre-registration. So thank you again, Roni, for speaking with us today and sharing your knowledge and wisdom. Roni will be speaking for 10 to 15 minutes, maybe slightly more, and then we hope to have a good amount of time for questions at the end. So if you do have questions, please use the Q&A box and we'll pass them on to Roni at the end. And thank you to Joe and Verena for, in the background, sending some links and things in the meantime. So I'll switch off my camera now and hand over to Roni. Thank you very much.
Thank you very much for the introduction. Let me just share my slides. So hello everyone, thank you very much for having me and for the introduction. Today we will talk about pre-registration in neuroimaging. Before we start, I just want to say some thanks. So first of all, to this team of people here who got me started thinking kind of seriously about pre-registration and how we can optimize it. I also want to thank Sophie and Antonia here. We worked together as part of a large collaboration that outlined good scientific practice in EEG and MEG research, and we co-authored the section on pre-registration. And before the talk today, they also shared some materials and thoughts with me, so thank you for that. And of course, the Credibility Neuroscience team at the BNA that does an amazing job in promoting these topics, educating scientists working in the field. I also want to apologize that this session will only be about human neuroimaging, but hopefully most things will be relevant to work with other species. Okay, so let's start with a very, very brief introduction to pre-registration. I assume all of you have some familiarity with it, but just to make sure that we're on the same page. So pre-registration is a time-stamped document that is publicly accessible or that eventually will become publicly accessible. And this document includes the hypothesis, data collection and analysis plan for a research project. And the key point is that it was written before the data were actually seen or analyzed. And as you can see here, over the last decade, pre-registration became increasingly popular, with more than 80,000 pre-registration documents on OSF alone. So in the webinar today, I'll talk about pre-registration in neuroimaging, but also about pre-registration in general, because there are many issues that are shared across fields, so we're not that special. I will share from my experience as an author of pre-registrations, as a reviewer, and also as a member of the community who just spent some time thinking about these issues. In the interest of time, I'll assume that you're already convinced that this is a good thing, and I will skip the why and why not. But I'm happy to discuss it later. I will then talk about the different platforms and how registration is done within these platforms. And then I'll talk about some of the things that you will need to consider in this context, including challenges, pitfalls, and recommendations. So let's say that you've decided that you want to pre-register your study or your project. Where would you register it? So there isn't really a right or wrong answer here. It depends on the timeline, the goals of your project, and etc. The first option is what we termed unlocked pre-registration. This means that you put your plans out there, can be in some internal repository or by presenting a pre-reg poster in a conference, for example. And the idea is to get some feedback on your plans before you actually register the study. I think that for neuroimaging studies, where the pipelines are so complex and there are so many options, that this can be especially useful. And it's important to note that this can be treated as a preliminary phase or stage, where the plans can still be changed. And then once you're happy with your plans, you can lock them with other pre-registration mechanisms. The second option is the public repository. The most popular one is OSF registries, which is part of the Open Science Framework. Once you posted your documents, you can immediately start running the study. And then when the study is completed, you submit it to a journal, and you should specify that the study was pre-registered and provide a link to the pre-registration. Some journals will explicitly ask if your study was pre-registered, but even if not, then you should include this information. The third option is to submit a stage 1 registered report to a journal. So you submit a protocol that goes through peer review and can receive an in-principle acceptance, or IPA. You then run the study, and when you're done, you submit a stage 2 report that will also be accepted as long as the plan from stage 1 was followed, independent of the results. The final fourth option is to pre-register with peer community in registered reports. Out of the 4 options, this is the only one that I haven't tried myself yet, so I cannot speak from my personal experience. The first steps with this are basically the same as for pre-registered report with the journal. So you submit your stage 1 to PCI, get it reviewed and endorsed, and then submit your stage 2 and get it endorsed as well. But now there's a twist. So there is a list of journals which are PCI friendly. And once your publication was endorsed by PCI, you will receive a list of journals for which your study is eligible. And you can choose where to publish your paper. And at this point, the choice is entirely up to you. The journal is committed to publishing your paper without any additional peer review. So you should notice that journals can specify additional criteria, for example, alpha lower than 0.02, for example. So if you want your study to be eligible for a particular journal, you need to make sure that you comply with the requirements before you submit your stage 1 report. The list of journals is currently quite limited, but it is getting longer. So, for example, Cortex, which was basically the journal that invented registered reports, does no longer accept registered reports directly, only those that were received through PCI. So then after you decided which platform you want to use, you need to actually pre-register your study. So if you go on OSF and start a new pre-registration, you'll see that they'll have several templates that you can use, or you can also go with open-ended registration or with a template that is not listed here. So it's quite flexible. Most templates will include many bullet points that you'll need to address and specify all the parameters of your study. Now, depending on your field, what you actually need or want to pre-register might change quite dramatically. And many fields now have templates that are tailored to specific protocols that are used in the field. So within our field, these are templates for fMRI and for EEG ERP. To my knowledge, these are the main ones that are relevant to neuroimaging in human, but I'll be very happy to find out that I was wrong, and please let me know if I am. The fMRI template was initially posted by Jessica Flannery in 2018 and was updated based on input that was received from many other colleagues. It's very comprehensive and it's relatively easy to follow. The ERP one, again, reflects a large collaborative effort and was posted recently, just in June 2022. It's also very comprehensive, and what I like about this one in particular is that it gives you examples for each section and every parameter that you can think about, which is quite useful. Now, if you want to submit a registered report, then in most cases, you won't be asked to use a template with bullet points, but instead, the format of your stage 1 report will look more like a journal publication with introduction and methods, but without the results and discussion, obviously. The guidelines are quite similar across different journals, with some variations in specific parameters, for example, the required power or alpha. My general recommendation regarding the format is that if you need to choose between a template and something that looks more like a paper, then try to do both. So work through a template to make sure that you tick all the boxes. and then write the introduction and methods like you would for a paper. So I'm currently working on the stage 2 manuscript for a study that was accepted on stage 1. And there is, I would say, a very particular kind of joy in just changing the tense from future to past and then have your study or your manuscript written already. So now we know what to do. Let's talk a bit about the challenges. And I think that the biggest issue that we had, at least, was to set a sample size that will be both realistic and informative. And this is an issue that can be relevant across fields, but I think that in neuroimaging, it is particularly challenging because the collection of neuroimaging data can be both expensive and time-consuming. And because of that, preregistration in the field can be some sort of a double-edged sword. So on the one hand, because it's so expensive, you want to maximize the chances that the results will get published, no matter what they are. You don't want to just bin the project later on. And preregistration increases these chances. But then on the other hand, you might have to commit to a very large sample size, or the required sample size might not even be feasible. To give you an example from our stage 1, the design is quite expensive with 2 fMRI sessions for each participant and also some behavioral testing. And that particular journal required a base factor of 10 to conclude that a result is conclusive and 95% power. And it turns out that with our specific design, we would need up to 100 participants to power for our hypothesis, but up to 440 participants to power for the null. And this was a sequential design, so we knew that we might not have to run all of these participants eventually, but we still needed to maximal N to be within the feasibility limit. So for us, 100 was still kind of within this limit, but 440 was way out of it. But because we did have quite specific predictions that we will find an effect. So after some back and forth with the editor, we agreed that we will power just for our predicted results and not for both the predicted and the null. But this goes to show that that really things can get quite complicated. And adding to these complications is the fact that with neuroimaging, it's really, really hard to determine effect sizes based on previous literature. And also that sometimes you want to test more than one hypothesis with your design. And then if you're using a sequential design, then it becomes very tricky to decide how to set your stopping criteria. I think what we definitely don't want is for preregistration to become a privilege that only rich labs can afford. And so we need to spend some time thinking about these issues and how we can make preregistration of neuroimaging studies more feasible while not compromising on quality. Another issue is that no matter how hard we try, some things might still remain subjective. So for example, when manually rejecting ICA components and other steps that are manual or semi-automatic. There are several initiatives in the community and community efforts to automate everything. Some of them are quite successful, but I think that there are also costs to that because if we don't do anything manually and not really forced to look at the data as it goes through different pre-processing procedures, then our understanding of the data is also reduced. In general, I don't think that there is a problem to include some manual steps in the planned pipeline, even if they're somewhat subjective, as long as we apply them in a way that is blind to the experimental conditions, and as long as the data quality is then estimated using contrast that is orthogonal to the contrast of interest. A third issue that is often raised is, as a concern, is that this is a strict format with no flexibility. So to this, most people in the community would reply that pre-registration is a plan and not a prism. And it is true to some extent, but it's also true that the flexibility is reduced. And this might be especially challenging with neuroimaging data. This is very complex and requires many steps of pre-processing with many parameters that can be tweaked in each step. And I think that one issue with the current format is that an analysis can be either registered or exploratory if it was not registered. But there could be several levels of exploratory analysis. So it could be that the data revealed an unpredicted pattern, and now I want to chase it further by running new analysis. And in this case, the analyses are exploratory in the sense that they are testing new hypotheses. But it could also be that I registered a certain pipeline and now want to try 10 other variations just to make sure that the effects that I get remain across all of them. This might not be something that I plan to do, but it's still confirmatory in a sense that it addresses my initial hypothesis. So I think that the current format does not really include space for this distinction. So yes, you could still do whatever you want within reason, even if you didn't plan for it to begin with. So the format can be flexible, but how these additional analyses are then linked to our initial predictions is not always clear. And then finally, an additional challenge is that it is time-consuming. So here I want to draw a distinction between two potential reasons for delays. The first might be that more time is devoted for planning. And this is not actually a problem, it's a feature. We should spend as much time as needed planning our projects. And if registration requires us to do that, then this is actually a good thing. The second reason, which is relevant to pre-registered report, is potential delays in the publication process. So some people will tell you that it shifts some of the heavy load from the publication phase to the planning phase, so eventually time difference should not be massive. However, the fact that registered reports need to go through the publication system at least twice, so once for stage 1 and another one, and once again for stage 2, and between these two time points, things might happen. And this is especially relevant to neuroimaging studies in which data collection often takes a while, and therefore these two time points might be further apart. So for example, a reviewer who reviewed stage 1 might not be available for stage 2. And from an editorial perspective, I can tell you that many times the bottleneck in the review process is not the completion of the review itself, but the ability of the editor to recruit reviewers. So if this needs to be done again, then it can lead to serious delays. So if you can afford to wait, registered reports are a really good option. If not, you may want to use another alternative. So finally, some recommendations to finish up. Start by going through relevant examples. I listed some here together with other resources and also the slides from this talk, which will also be available through the BNA website. Make sure to pilot your paradigm. This will help you to know what to expect in terms of setting exclusion criteria, defining outliers, and so on. And will also give you a chance to develop your pipeline before you start collecting the full data set. Try to have concise hypotheses and to clearly link them with specific patterns of results. It's useful to have one key hypothesis that will be used for power calculation and to determine the stopping criteria. Try to get as much feedback as you can. If you go with registered reports, then this is kind of built into the system. But try to get feedback even if your pre-registration does not go through peer review. I would also suggest collaborate with colleagues, especially those with complementary skills. They can give you useful feedback and will also allow you to make sure that all the aspects of your projects are covered. Now, this is kind of a general advice, I guess, not just for registered reports. For registered reports, if you realize that you need to significantly deviate from your accepted stage 1 plan, make sure to discuss this with the editor before you implement any changes. There might be some consequences to the deviation that you didn't consider, and it might result in the rejection of your stage 2 report if you don't get these changes approved. So that's all from me. Thank you very much.
Thank you so much, Roni. That was a really thorough top line on a lot of the issues, and also with some great specific advice for neuroimaging. So We've got just 10 minutes for questions. We've got, please do put your questions in the Q&A box. We've got a couple waiting in there. And perhaps while people are thinking, just 1 point, 2 points that I picked up on was you really, you made a really important point there about doing or ensuring that registered reports as a system are accessible to everyone. So for example, by some journals mandating these really high base factors or something that makes it unfeasible for others. And also you said, like, if you can afford to wait, then it's a good thing. So there's lots here which is talking about high and low resource areas. So is there anything that the people who are looking at these things and trying to champion it, is there anything that they're doing to address these things, or are they being considered in ways?
Yeah, so I know that at least in my university, preregistration has become part of the training that we provide to students. So this might help a little bit with some delays or hiccups that result just from not knowing what to do or how to approach this at all. What I think would be really helpful in the sense is to kind of integrate this with grant applications. So one catch with registered reports, for example, is that you need to show that you have the funding resources already to run the project when you submit your stage 1 report. Right. But how nice would it be if we could just submit the report and then get funding based on a project that we already know is going to be published later on. I think this can solve a lot of problems, especially in like allowing less privileged labs to also have access to this.
Lovely, thank you. And I guess a point we can all take is if you get asked to be a reviewer for a registered report, maybe if we get asked to be reviewers from registered reports, perhaps that's something that we could look on favorably, knowing that this is a move to try and improve access and equity and things. Great, thank you. So I've got so many questions for you, but I won't steal all of the airtime. So I'll take the questions from the Q&A in the order they came in. And the first one is, do you know of any completed fMRI registered reports?
Yes, so there was a QR code with a link to OSEF. I'll share the slide. I can share it again. I put some examples there. I think 2 of them are fMRI, completed fMRI projects. One was pre-registered with OSF, and I think the other is an fMRI study that was a registered report. So yes, there are some.
Someone has done it.
Yeah,
great, thank you. So our next question
hopefully
is,
ours will be available soon, but yeah, still in progress.
We'll all be watching and waiting. So next question, if the pre-registered report is rejected, could I try to publish the article once it's completed, or will it decrease the chances of getting accepted? So I guess I think they mean if the registered report is rejected, so you try to register a report, it's rejected, and then you try and publish anyway.
Okay, so I think it depends on the stage. If you're asking about stage 1, then yeah, sure. If it went through a review and was still rejected for some reason, I would be very, I would try to kind of follow the suggestions of the reviewer and try to improve the design as much as possible for this to then be, publishable in another journal. As if it was rejected on stage 2, I think it's hard to say. Depends on the reason for the rejection, because normally a paper should not be rejected during stage 2. So this would be either because the authors decided to withdraw the paper because they found out that there are some severe problems with it, or because they deviated from the original plan. And then in this case, yes, you could go to a different journal, but there will be a trail. So the journal that this was retracted from will still have the stage 1 available, and any deviation, it will be out there. So just keep this in mind if you decide to then publish in another journal.
That's great, thank you. So our next question, This one's slightly longer, and I'm dyslexic, so reading off the cuff here. So how would you go about pre-registering a study with clear research questions / hypotheses, but no clear specific methods yet of analysing them? I.e., there are multiple sound analysis options, for example, many ways to select ROIs. Would you put the options down in the pre-registration and state that this is exploratory, or spend more time piloting to pick an option and state that? So do you put more kind of potential analysis paths in your pre-registration, or spend more time piloting and wait until you've settled on a solution?
Yeah, so I would do two things. The first thing I would do is to do the self-pre-registration thing. So try to get feedback at the stage when this is not locked yet. Just get feedback from colleagues. Maybe they will have any recommendations about what is the best pipeline to go with. The second thing, yeah, definitely pilot as much as possible. This is really important to just pilot, both to see that you get a behavioral effect and also that your analysis pipeline is suitable. And then I had another thing, I can't remember it now. Yeah, so I think, yeah, so your question about whether to state this as exploratory analysis, this comes back a little bit to what I mentioned about the distinction between confirmatory and exploratory. So in principle, you're not supposed to mention any exploratory analysis in your stage 1 protocol. But sometimes you already know in advance that there are things that you would like to do. So I think this is where this format is not entirely flexible, doesn't really give space for everything that we might like to do. And I think that this is definitely something that we can improve with this. But yeah, definitely pilot if you're not sure.
Great, thank you. And I'll just tag in another question on my own here because it was related. So you mentioned that it's okay to optimize your analysis pipeline as long as you're blind to the conditions, like the experimental conditions. So I know that a lot of imaging studies, the same person who wrote the stimulus also collected the data and also is analyzing it. So I know it can be hard to sort of differentiate, have someone doing the blind analysis, blind of conditions, but I think that's important. But how would you describe that in your pre-registration? So linked to the previous question, how do you describe the potential optimization that you might take and state that you're going to be blind and see which one of these works best?
Yeah, so I think even though we know what conditions we implemented in the study, then it can be quite easy to be blind to that because most of the pre-processing steps occur through all the data, right? So, for example, with EEG, you don't analyze each trial separately, knowing what was the trigger for that, or you just do it for all of them. So I think that this can be done. And I think just in the pre-registration, just mentioned that some steps will involve some manual procedures or semi-automatic procedures, and that the person who is analyzing this would be trained on how to perform this, or is already trained.
Yeah, I guess the point is that don't get all the way to the end of your GLM and then decide which optimization was preferential.
Yeah,
like
exactly.
It needs to happen before.
Yeah, what I was referring to is to kind of cleaning and pre-processing stages that occur before you actually look at the experimental conditions or the averages or whatever. Obviously, this shouldn't be done. So once you know the results, you shouldn't go back and reanalyze just to, but any preliminary steps that occur before that, then they can involve some subjective decisions, or hopefully these are objective decisions, but we're all people. So
absolutely.
Yeah.
So we are just at time now. So if anyone needs to drop off the call, then you're very welcome to. Thank you so much for joining us. There's one more question in the Q&A that I'd like to put towards Roni, if you're okay to wait just one more minute. And before everyone leaves, I just need to mention that this is the first of 3 lunchtime seminars. Our next one will be about data privacy, and then we have another one around the middle of February on some other thing, which I can't remember what it's on now. I'm sorry for that. So do look out for the next webinars, and we hope we see you again to the at the next one. So this last question in the Q&A is, how would you recommend that we incorporate registered reports during a doctoral program? As we know, graduate students, timelines are very tight and milestones are very defined.
Yes, that is really challenging. So one option would be to go with pre-registration that doesn't actually entail any delays, because you don't need to go through the publication process twice. So that would be one option, which is perfectly valid, and I think PhD students should consider this. Another thing to do is to maybe, so like if this is an established lab and there is another person in the lab working through a similar pipeline, then things can be slightly reversed. So once you join the lab, start working with another person on their results that were already, or data that was already acquired, while developing your own project.
So thanks again, Roni. This is a controversial, it can be a controversial topic, because we know how much us imagers like our analytic flexibility and our degrees of freedom. But so I think it's a really interesting, like, cultural behavior change issue to see how we can make registered reports work for us and our PIs and our timelines and our funders. So thank you for sharing your knowledge on that again. So I understand that slides are going to be made available afterwards, and I'm sure Roni would be happy to be contacted by anyone with further questions. Great. So thank you again. Thank you to BNA for hosting us. I look forward to seeing as many people as possible same time next week.